Hello, everyone. Thank you for your interest in this talk. My name is Stephanie Gage, and I'm going to present a true team effort, and it is my pleasure to be the one to share our work with you. I'd like to highlight my co-authors, Brett Alio, Varun Sharma, Jordana Sprayberry, and Simon Sponberg. This is a new project for us and one that we are excited about. Our talk is titled, Does an Ecologically Relevant Odor Influence Visual Motion Selectivity in the Hawk Moth Nerve Cord? To the left is an early naturalist illustration of Manduca sexta, the species used in the study from 1828. So our nervous systems are tasked with keeping us alive during changing conditions. And there are numerous examples where animals may respond to a stimulus differently, depending upon the context or the behavioral state of the animal. For example, in humans, we may respond to our favorite food differently, depending upon whether we ate a large meal 10 minutes ago or we haven't eaten for several hours. In other examples, a mother polar bear may experience heightened awareness if her cubs are present so that she can detect danger at the earliest indication. Or, as any beekeeper can tell you, when a honeybee hive becomes more irritated and defensive than normal, that may be a sign that they lost their queen. At the level of the nervous system, how might contextual changes like these be encoded? We've learned a lot over the past 20 years, especially when it comes to visual processing. The function of a visual system is to condense and transform patterns of light into a form that is meaningful to the rest of the brain that could also be propagated to enable movement. One important aspect of this transformation is to emphasize behaviorally relevant stimuli and de-emphasize less important ones. A classic study from McAdams and Monsell in 1999 found that attention can modulate visual processing. Primates were trained to either pay attention to the stimuli in the receptive field or to other stimuli outside of the receptive field. In this way, orientation tuning curves could be constructed from two different behavioral states. What they saw in the macaque V4 was that attention could increase the gain of the response, but it didn't change the orientation selectivity. If we consider the analogy of a radio, attention turned up the volume on the station that you were listening to, but it didn't turn the channel to a new station. Similar results were found with locomotion in mice. When transitioning from standing still to walking on a ball, evoked visual responses were more than twofold stronger. The preferred orientation of V1 cells were not significantly altered, but it was as if the volume or the gain had been turned up with locomotion. And there's a similar story in flies. Walking not only increases the gain, but also boosts stimuli at higher frequencies, which causes the tuning curve to change shape. The functional significance of this shape is still unfolding, but because the peak of the curve shifts towards higher frequencies, this modulation may help flies process faster visual motion. In other words, the act of walking enhances the speed in which the background moves, necessitating a need for faster visual processing. In this case, walking turned up the volume and may perceive that the song is playing faster. And there have also been similar findings in the last 10 years, particularly in Drosophila. Similar effects have been found in all three areas of the optic lobe. So there appears to be a consensus emerging. Visual processing is highly modulated by behavioral state. We typically see an increase in gain and or a shift in frequency tuning towards higher velocities. And so the question that we have here in this study is what transitions an animal into the internal state underlying attention or locomotion? Can an external cue, like a relevant odor, produce similar changes in the tuning curve? So in this study, to better understand this broader question, we ask, does the addition of an ecologically relevant odor similarly affect visual processing? In other words, can an external sensory cue produce changes similar to locomotion or attention? We know that for many animals, insects especially, an odor from a food source or a mate can propel the animal to move to go find these sources. So we may expect that presenting an odor may incite the internal state of locomotion. So as our main hypothesis, we expect that an odor will shift the tuning curve in similar ways, both increasing the gain and shifting the responsiveness towards higher frequencies. In addition, we also expect odor to improve the precision or phase locking of oscillating flower motion responses. Once the source of the odor has been located for an animal, it's advantageous to better feed from a flower blowing in the wind or to locate a moving mate. So to test these questions, we use the hawkmoth Manduca sexta. Manduca relies on both visual and olfactory guided flight to locate food sources, to lay eggs, or to find mates. And because of this reliance on both visual and olfactory information, and the fact that both sensory modalities have been well studied in Manduca, we could ask the question of whether an odor modulates visual processing. Now, Manduca feed from Solanaceae. You may find them on tomato or tobacco plants, or if you're in the Southwest on Datura radii. Datura produces these elongated flowers that open after dusk. This connection between Manduca and Datura is thought to be co-evolved. The odor produced from datura flowers produces innate responses in Manduka. In the lab, we use a 7-cent datura blend, which serves as our ecologically relevant odor. Now, to test our hypotheses, we are presenting two types of visual motion. One is wide field motion, or the background visual seen as the moth navigates the environment. We presented left and right motion to the moth at varying temporal frequencies, ranging from 0.1 to 30 hertz, and we kept the spatial frequency consistent at 0.03 cycles per degree. In addition to wide field motion, moths need to track near field moving objects like a flower as they move with the wind in the environment. We presented an oscillating flower stimulus to gauge object motion tracking responses at the range of frequencies from 0.1 to 10 hertz. So we chose to go downstream into the ventral nerve cord, or the VNC, to record activity. The left picture shows the exposed VNC. We then insert a 16-channel multi-electrode and play both types of stimuli on the screen in front of the moth. Now, the reason that we chose the VNC was that wide field motion and oscillating flower responsive units have both been found here, thanks to folks like Brett and Jordana's work, as well as Sanjay Sani's group. The VNC functions as a bottleneck. It relays sensory information from the brain and motor information from the thoracic ganglion. To answer our question about whether an external cue like an odor can set the change in responsiveness similar to movement, we chose to look here in the VNC. Now, we created our experimental design with the light cycle of the moths in mind. Hawk moths are crepuscular, and one of their most active times is between 1.5 to 2.5 hours after dusk. So we began our experiments at this time. We first began with our pre-odor condition by presenting 2 trials of each stimulus type. Then, with our datura odor on, we present 2 trials of each stimulus for a total odor duration of 17 minutes. We then test again at 10 minutes and 60 minutes after the odor is off. Now, what do these responses look like? Now, these are wide field motion traces at one individual frequency, and this particular trace suggests odor modulation. Now, if we look at our before odor trace, we see that as the stationary stimulus appears on the screen, we see some activity, followed by more activity when the stimulus moves. We see 3 different units, and each raster mark indicates when that unit fired. So we see that when odor is applied, we may not see a large change in unit 3, but we do see an increase in responsiveness in unit 2, and unit 1 actually becomes active with odor. We then return to our pre-odor baseline at 10 minutes post-odor. So how many units look similar to this? So here's a breakdown of the responses. We find that the majority of our units show an increase in responsiveness to odor between 51 and 55%, depending upon whether they respond to left or right moving motion. And the opposite is also true. We see a decrease in responsiveness to odor between 10 and 26%, and some units show no change. Now, to answer our questions about how an odor may be similar to locomotion, we need to visualize our data with a frequency tuning curve. So on the right is an example of a tuning curve taken during one wide field motion stimulus presentation. Here we see 3 units that are responsive to this motion in red, peach, and pink. And you see varying responses at different frequencies. But this is only one stimulus presentation. We don't know how the red unit, for instance, may change during the course of an experiment, particularly when odor is on. So we need to visualize our data to see how odor responsive units change as a whole. And what we find is that odor modulates the frequency tuning curve. In red, we see our tuning curve without odor, which is an average of our 44 units. And when odor is applied, we see the following in the blue trace. We see an increase in the gain at multiple frequencies, meaning there's an increase in the spike rate when odor is applied. And the largest changes occur at 1 to 3 hertz. We also see a shift at the top of the tuning curve. The peak has shifted 1 to 2 hertz towards faster frequencies. So what we see here are similar changes to the ones we saw earlier with Drosophila and the effects of locomotion. There is an increase in gain and a shift in peak. And there is one other interesting element on this graph. We see the effects of aliasing with odor. At very high frequencies, we see that the odor modulated curve dips below 0. This is the aliasing effect, where the stimulus is moving so fast, the object appears to be moving in the opposite direction. This is the first time we see this in the VNC. Because it only occurs with the odor condition, it may be behaviorally relevant. So to come back to our questions, can the addition of an ecologically relevant odor affect visual motion processing? The answer is yes. The next question is, how might an odor affect frequency tuning, and is it similar to prior studies of attention and locomotion? And the answer there too is yes, there is an increase in the gain and a shift in the peak towards higher frequencies. Now, lastly, can odor modulate the precision or phase locking of flower motion responses? So before we get to precision, I want to share with you the odor modulates oscillating flower responses as well. We see that 26% of our units are positively odor modulated, 37% are negatively modulated, and some show no change. And in these plots, you see the difference between positive and negative odor modulation. On the left, you see a sinusoidal wave, and you see the responses to the oscillating flower before odor, during odor, and post odor. We see an increase in the number of responses during odor presentation in green, and this response returns towards baseline. On the right, you see the opposite. The unit disappears during odor presentation. We see that the green dots go away and returns post odor. Now to return to our question about precision. And I'll tell you what we mean by precision. So this video shows you again our oscillating flower stimulus moving from left to right. And on our sinusoidal wave, the left and right motion corresponds to these phases. Now, how do we measure whether odor may affect precision? So we can measure precision by calculating vector strength. Vector strength is a measure of how phasic responses to floral oscillations are, or how precisely responses track flower motion. So an example of lower vector strength would be a unit that fires similar to the red trace. The spikes are not well aligned to the phase of the sinusoid, but occur intermittently. Now the blue trace would be higher vector strength because the firing corresponds more with phase. So ultimately we're asking, does the addition of an odor increase or decrease the vector strength? In other words, does odor affect the precision or phase synchrony? And what we find is that odor modulation does indeed change the precision of flower motion responses. This graph shows normalized average vector strength throughout the course of the experiment. We see increased precision in the positively odor modulated units and a decrease in precision in negatively modulated units. Some units return to baseline 10 minutes after odor duration, others 60 minutes. So we do see a jump in activity in some units at 60 minutes that exceeds what we saw previously in the experiment. So to return to our last question, can odor modify the precision of oscillating flower motion responses? And the answer is yes. We see both increased and decreased precision with odor depending upon the unit. So most importantly, what do these results mean for the animal? An ecologically relevant odor may function as an external cue or behavioral switch that prompts the moth into an internal state similar to or in preparation for locomotion. This could be why we see changes similar with an odor that was previously seen with locomotion or attention. In addition, their change in gain and temporal selectivity suggests that odor may function as a fine tuner to a moving visual scene and to enable more precise object motion tracking. And with that, I'd like to conclude our talk and thank the members of the Spanberg Lab for all of their support, encouragement, and feedback. Thank you.